Great, let's get started. All right, I'm still kind of hashing out how far I think that maybe we'll get before the next exam. The next exam's not next week, it's the week after. So kind of on the horizon. I'm anticipating we'll at least get through all of our genetic stuff. I don't know, we'll see if we get started on viruses or not, but at least through chapter 7, I hope. So last time we just started, we finished up metabolism, talked about the difference between anaerobic respiration and fermentation. Kind of went through the rest of respiration. Just started with kind of structures of DNA, and we talked about kind of big picture how DNA is replicated. We know, thanks to Messelson and Stahl's very eloquent experiments that we kind of went through kind of quickly there, that DNA is replicated semi-conservatively, meaning one old strand stays in the new daughter strand. So you get an old strand and a new strand. One strand is conserved from the original double-stranded DNA. Okay, so are there questions? Don't be afraid to jump in and ask questions. Give you a chance to think about that a little bit. So let's now think about this polymerization reaction of how a DNA strand, how a new one is made from an old one. So we'll kind of look at it, kind of zoom in, but it is a polymerization reaction. We get not just nucleotides being added on to make a strand of DNA, but actually they're triphosphate nucleotides. So what that means is that the nucleotides, so remember the deoxyribose and the base together, that's considered a nucleoside. All right, if we count the base, in this case guanine, along with the deoxyribose here and a phosphate, that's a nucleotide. But then when there's actually two more phosphates here attached, that's a guanine triphosphate deoxyribonucleotide. That's kind of a mouthful. It's a little bit cumbersome, but that's what that DGTP stands for. That nucleotide actually carries with it the energy, two more phosphates, to power that polymerization reaction. Okay, so whether it's adenine, guanine, cytosine, or thymine, okay, those again, triphosphate nucleotides are floating around in the case of bacteria in the cytoplasm of the cell. Okay, so when those are added on to an existing DNA strand, okay, and there's going to be the old strand over here, there's going to be a template so it knows which ones to add on. Here's a triphosphate nucleotide. It's always going to be adding on to the 3' hydroxyl group of the new strand. Okay, those two phosphates get cleaved off. Y2 instead of 1. 2 makes it extremely energetically favorable. So that reaction happens really quickly. And then you've got that new guanine added on to that old strand, new strand, sorry. Okay, so moving forward, there's lots of different enzymes that we're going to talk about that facilitate this. Okay, so I want to start out just giving you a a pre-question, like before we talk about it. Okay, so take a minute, read this over. I don't expect you to know the answer. Maybe you talked about this in another class and you're familiar with all these enzymes. Read it over and just make a note of which one that you think is correct. All right, so jot it down or tell your friend which one you think it is. I'm not going to tell you the answer yet. And we're going to go through replication, and then I'm going to put it up there again, and we'll talk about, by that time, I bet you'll know which answer is correct. So jot it down, and we'll see. If we were doing clicker questions, this would be more fun because we could see, but I can't record and do clicker questions at the same time. So, okay. All right, so hold that thought. Okay, so DNA replication. We are always talking about how this is happening in bacteria. As we talk about replication and transcription and translation. At times, I will point out differences between what process we're talking about and what would happen in humans. But I always assume that we're talking about bacteria. In bacteria, so in replication, all the DNA gets copied. So in my mind, replication is just a little bit simpler than transcription and translation because you don't have to figure out how it starts and how it stops. Basically, just everything gets copied. So for DNA replication, that starting point, we do have a name for it. It's called the origin. And a lot of times we just kind of skip over that name because you know what that means. It means it's when something starts. It's the beginning of something. So it's something that's familiar already. That's, again, the site for the start of replication. Okay, prokaryotes typically are only going to have one origin. So in this picture down here, that little green square, that's the origin. How do the enzymes involved know that's where to start? That's a good question. There's probably some methylation that marks that spot. There's probably sequences of A's and T's. Remember, those A's and T's only have 2 hydrogen bonds, so it's a little bit easier to pull those complementary base pairs apart. There's probably some other signals involved there. Eukaryotes will typically have many origins because eukaryotes have way more DNA to replicate. So you may have seen multiple, may have seen this sketched out somewhere. They will have multiple replication forks opening up in their DNA because otherwise, if they only had one, it would just take too long for all the DNA to get copied. Okay, so prokaryotes, less DNA. Remember, they only have one chromosome typically for prokaryotes. Okay. And then the important enzyme involved here to begin with is helicase. All right, so helicase is going to basically break those hydrogen bonds between the complementary base pairs. Sometimes we say, we describe that as unzipping. So it unzips the DNA strands. I've also heard that referred to as melting the DNA, but kind of the term that we always have used in the past is unzipping, pulling them apart. Okay, so you can see down here in figure 7.7, if we have two replication forks opening up at the origin, one going one direction and one going the other direction, the helicase enzyme there is working here and here to unzip those complementary base pairs. Sometimes we call this a theta structure because it looks like a Greek theta here and here, but it's just, you know, replicating that round, a circular chromosome replication going in both directions until we end up with 2 chromosomes, okay, or 2 plasmids or whatever that is. Okay, so going in both directions, the helicase helps facilitate that. Okay, there's a lot more enzymes involved, but if you only remember one, the most famous one is this DNA polymerase 3. Okay, if it's the most famous one, why isn't it DNA polymerase 1? Well, the answer is they were named in the order that we discovered them. So I don't know, for whatever reason, this was the 3rd one discovered. But DNA polymerase 3, this is the enzyme that's adding those triphosphate nucleotides onto the existing strand. Okay. Okay. So thinking about direction, okay, that's always something that we want to sort of be aware of. And this is shown a little bit better on the next slides. But that DNA polymerase is moving this new strand. Remember, the DNA strands are always anti-parallel. So the new strand, this is going to be the 5' end, and this is going to be the 3' end. Okay, so if we know that it's moving in this direction towards the replication fork, then we know that this is the 3' end. Okay, this enzyme always has to move along the old DNA in a 3' to 5' direction. So that means on the opposite strand, it's going to be going in this direction. All right. Okay, so I think that's sort of the confusion sometimes about the directionality of how this happens, whether you're talking about moving along the old strand or the new strand. Okay, it's going to be opposite. If you can remember, it's always adding on to that 3' hydroxyl group, then that's really all you need to know. Okay, so it's always adding on to that 3' hydroxyl group. Okay, this is a pretty special enzyme. It moves pretty quickly. It's going to add somewhere between oops, erase that, 500 to 1,000 base pairs per second. Okay, mind-bogglingly fast. The number of nucleotides, triphosphate nucleotides that it can add on there. And it has a really good proofreading function. We'll say great proofreading. So in other words, if it accidentally adds a nucleotide that doesn't match up, that's not complementary with the template strand, it can kind of look behind it and check and make sure it put the right one in. And if it didn't, it will fix it. Okay, so good, really great proofreading. How often does it make a mistake? It's 1 in 10 billion base pairs will it make an error. Okay, which is what we want, right? This is the blueprint for the cell. If you make a mistake in the blueprint, then everything that's made using that instruction is always going to be wrong. Okay, so we need the blueprint to be correct. Okay, if you write down your recipe wrong, every time you make that recipe, you're going to make a mistake. So it's important for the original instructions to be correct. Okay, and also you'll notice here in this picture, here's the DNA helicase. That's the unzipping enzyme that we just talked about. There's also some stabilizing proteins there that just help keep those 2 strands apart so that the enzymes can work. Okay, so if we're talking about first the leading strand, so that means when the DNA polymerase 3 is moving in this direction towards the replication fork, really there's not too much more to say here other than that DNA polymerase can't bind there and just starts making new DNA by itself. It needs something to start from. So there has to be a primer involved there. There has to be this 3' hydroxyl group for the DNA polymerase 3 to actually add the new strand on. So we need an enzyme called primase to come in and synthesize a temporary RNA primer. Okay, this is going to be somewhere between 10 and 20 base pairs. So a really short stretch of RNA, and that's going to be temporary because you can't have little bits of RNA mixed in with your DNA. So that's just going to be temporary to get everything started. Other enzymes are going to come in and get rid of that and put in some DNA instead. Okay. So that happens at the origin, and then, you know, again, it just proceeds. Okay, so that's pretty easy. The opposite strand is what, so that's the leading strand. The opposite strand is known as the lagging strand. Okay, so this is where we have to have a series of bits of DNA done in order to get the RNA polymerase moving in the right direction. So we have a series of fragments that we call Okazaki fragments. Because again, here, 3' end, we're always adding on to that in that 3' direction. Okay, so you have a series of primers instead of just one primer. Okay, so here's one here, they're in red. Here's one here, here's one that's just being removed. Okay, you have a series of primers. The DNA polymerase 3 adds the triphosphate nucleotides up until the next strand. So it's kind of going like this, here, and then here, and then here, and then here. These Okazaki fragments are somewhere between 1,000 and 2,000 base pairs. Okay, so segments. Okay. And then let me write this in. The DNA polymerase 3 synthesizes the DNA. But then we have to get rid of the primers. So another enzyme comes in, DNA polymerase 1. You can see that right here. It's going to get rid of the primers. And by get rid of, I mean it chews it up and then synthesizes a little stretch of DNA in its place. And then one more enzyme that we're going to mention, and that's ligase. And that's pictured over here. There's just a little something extra that's needed to kind of seal up the gaps. Okay, so it basically adds some bonding to just make it, you know, a nice smooth line of nucleotides. So so add some bonds, we'll say, to kind of seal the edges. It's actually a phosphodiester bond. It has to add a phosphate in order to make it intact. Okay, so there's a lot of enzymes at work there. You're going to want to understand what all those enzymes do. Here's a short video clip that I thought was pretty good because it really just mentions all the enzymes that we're kind of interested in at the right level. So I forgot to post this in a way that I wouldn't get ads. So let's see if we get an ad here. Oh, yeah.
Treat yourself to rich Ghirardelli chocolate. and luscious
Just a
caramel.
short one. Okay, so this is just like a minute and a
When
half
DNA
long.
replicates, its strands are
Shows the
separated by the
DNA,
enzyme
so
helicase.
here's the helicase enzyme that's going in and breaking those bonds between the two strands.
Single-stranded DNA binding proteins keep the strands from reannealing.
The little orange circles are the stabilizing proteins.
One DNA strand encodes the leading strand, which forms from its 5' to its 3' end, using DNA polymerase 3.
Making the new DNA.
No problem here, but the lagging strand presents problems. It has to form from 5' to 3' too. It forms in pieces called Okazaki fragments.
So the lagging strand, it has to make Okazaki fragments.
First, an RNA primase lays down an RNA primer. Then, DNA polymerase 3 lays down new DNA.
Another DNA polymerase 3 making the new DNA.
The process repeats again and again.
See that in slow motion, just in case you missed that.
DNA polymerase 1 replaces the RNA primers with DNA. Finally, DNA ligase links the Okazaki fragment.
It's kind of nice animation there again with all the vocab that you're going to want to know for that. All right, so just a couple more things to mention about replication. There's another enzyme that's really important because if you look at our picture over here on the right, as the DNA is unwinding and unzipping, as we're pulling those two strands apart, eventually this causes kind of upstream here some twisting, okay? And eventually the mechanical strain of that makes it so those two strands won't be able to come apart anymore. So you know this if you've ever, been swinging on a swing and twisted around, eventually you're getting down lower and lower. You can't pull those chains apart anymore. Okay, so there has to be some way to alleviate that. Here's a nice, illustration of two rubber bands, right? You're pulling them apart. You get this super coiling upstream. Okay, so we have to have a way to alleviate that. There's an enzyme in bacteria called DNA gyrase. And DNA gyrase does a bunch of things. That's kind of unusual. Usually enzymes will just do one very specific thing, but it actually will cut both the strands, unwind it to get some of those extra coils out of there, the super coils, and then seal the ends again. Okay, so this is an enzyme that's only bacteria have. We have to do that in our cells as well, but the enzymes are slightly different. It's different enough that we can have, there's actually a whole class of antibiotics that target bacterial DNA gyrase. Okay, fluoroquinolines. We'll talk about those later. So again, we have enzymes that just are slightly different. Okay, and then lastly, there is some DNA methylation that takes place. And I mention that now because we're going to come back to it later, but that just means this functional group, CH3, gets added onto the DNA at some point. Usually it adds onto adenine nucleotides. Eukaryotes, there's methylation that happens in eukaryotes as well, but it usually happens in cytosines instead of adenines. What's the point of it? There's several different functions. We think that it helps define the origin, the starting place. We know that it's involved in DNA repair, so we're going to talk about that a little bit later. What if bacteria have a lot of different mechanisms, enzymes that will move down the DNA and fix any kinds of errors that are there. If there's a mismatch error, meaning say there's a guanine paired with a thymine that shouldn't be, how does it know which one to fix? It depends on which strand has been methylated more. last. So the old strand would have been methylated already. Maybe the new strand wasn't methylated yet. So we'll talk more about that later. But there may be some regulation involved as well as, you know, DNA that's methylated may be more likely to get transcribed into messenger RNA, for example. So lots of different functions. Okay, so back to our question. Okay, so we just talked about this. The role of DNA gyrase, that should be answer D. All right. So if we go back and kind of think about, well, what's the answer to what's the enzyme that seals the Okazaki fragments together? I would say that should be ligase. And I know you don't have this question in your notes there, but the one that unzips the complementary bases, okay, that's the helicase. Adding the new triphosphate nucleotides to the growing nascent strand. You might have heard that term nascent. That just means the new strand, the new one that has potential. That's DNA polymerase 3, and then the synthesis of the primers, right? So you gotta want to know the role of all those enzymes. Okay, questions? All right, so taking just a little step back and again, thinking it big picture before we move on, thinking about the flow of information. So an organism's sequence of bases, right? There's 4 different bases. That sequence is known as the organism's genotype. Okay, and when one of those strands of DNA is made into RNA, in this case, messenger RNA, we call that transcription. And then the messenger RNA gets at the ribosomes translated into a chain of amino acids that we would call polypeptide. Okay, when that's long enough, we would call it a protein. Okay, and what proteins an organism can make, that determines its phenotype. All right, remember that's the observable traits. Okay, so you might have heard it often said, oh, an organism's genotype determines its phenotype. Right? There oftentimes may be some environmental factors at work there too, as to what genes get transcribed and which ones don't. For bacteria, what's an observable phenotype? It's always a little bit harder. We might have to do some tests in lab, but it might be the shape of the cell. It might be whether the organism can metabolize some sugar like lactose or sucrose. You know, lots of, you know, it can make this enzyme, it can't make that enzyme. So that's more when we're talking about traits, that's what we think about for bacteria. All right, so as we think about how DNA is made into RNA, let's first think about different kinds of RNA. Okay, so messenger RNA is kind of the first thing we think about. Let's first look at the structure of RNA and then the different kinds of RNA. Okay, so thinking about how RNA is different than DNA, of course, RNA is single-stranded. There is an exception to that. You might think of where in nature we might have double-stranded RNA. Okay, there are some viruses that have double-stranded RNA. Not very many, but there are double-stranded RNA viruses that infect humans, that infect plants, that infect other organisms. So just to throw that out there, but you know, in our bodies, in bacteria, it's always going to be single-stranded RNA. It has a primary, secondary, and tertiary structure, or it can. So what I mean by that is a primary structure is just the sequence of bases. Secondary structures are things like alpha helices or beta sheets, kind of regular shapes, consistent shapes that we often see. And then a tertiary structure would just mean some more complicated folding of the RNA. So we can find RNA that has all those different forms. It's going to have uracil instead of thymine. DNA has thymine, and it's going to have a ribose sugar instead of deoxyribose. So if you look up here at a ribonucleotide, it looks a lot like a nucleotide. Again, the ribose sugar, the only way that it's different from deoxyribose is that that second carbon, there's a hydroxyl group there instead of just a hydrogen. Okay, so that's the only difference between ribose and deoxyribose, in case you're interested. Okay, so really quite similar. All right, so types of RNA. I wrote a couple things in here because when I went over this before, I kind of forgot to say it to myself, so I wanted to remind myself. So we already talked about there are RNA primers. Okay, remember those are really short pieces of RNA, maybe only 10 to 20 base pairs that are helpful in DNA replication. In terms of total RNA in the cell, RNA primers make up a really teeny tiny small amount of the total RNA in the cell. Okay, almost negligible, and of course they're temporary. Messenger RNA, that's kind of RNA that most people think of because it's kind of when we're following the flow of information. But actually only about 4% of the RNA in the cell is messenger RNA because it's kind of fragile. It doesn't stay around very long. It makes sense if it stayed around for a long time and it just keeps getting made into proteins over and over and over again, the cell could be making proteins that it didn't really need. So it makes sense, transcribe the messenger RNA, it maybe lasts for just a little while. If it needs more of that protein that it's making instruction for, the cell will make more. Okay, so there were a lot of hurdles when, 30 years ago, we started thinking about using messenger RNA as, something that could maybe cure disease. There's a lot of hurdles to getting that cells to take that up because it is so fragile. And of course, lots of hurdles in using it as a vaccine as well. Messenger RNA in prokaryotes, we could describe that as polycistronic, meaning that one messenger RNA segment can actually encode for several proteins. Okay, that's the basis of operons that we're going to get to later. It's actually a nice way to regulate proteins together that have a similar function or that are all needed in one pathway. So kind of just translate them all together. Eukaryotes, though, are going to be monocistronic. Cistron means gene, so one gene, meaning so you get one protein from one messenger RNA. That RNA encodes for one protein. Okay. And then we have transfer RNA. Transfer RNA makes up about 10% of the total RNA in a cell. So transfer RNA are going to be vitally important when we talk about translation. Let's just briefly talk about it now, even though we're getting ahead of ourselves a little bit. We haven't talked about transcription yet. But they are the molecule that delivers the correct amino acid that matches up with the messenger RNA sequence. And that's going to be delivered to the ribosome. So here's your book's pictures of messenger RNA. Typically, we draw it looking like this in kind of a clover leaf form. That amino acid is going to be attached up here. And which amino acid gets attached depends on the anticodon, or at least not totally just that. There's other clues in kind of the shape of the molecule, but those three bases there, in this case, ACC, I think I looked it up, that will match up with the arginine amino acid. So that depends what amino acid. There's these hairpin loops, usually three of them there. Okay, so you can see kind of, it's not just a straight line, you can see some kind of unique structures here. It turns out that we think, you know, when these are working in the cell, they don't really look like that. They actually look like the picture on the right, how they're, you know, folded in kind of a complicated way, more of a tertiary structure. So when your book's talking about tRNA, they use this kind of upside down L icon just to denote tRNA rather than these kind of cloverleaf shaped picture. Okay, so we'll talk more about their role when we get to translation. And then there's ribosomal RNA. So ribosomal RNA makes up about 85% of the RNA in the cell. And it's the kind that everybody always kind of forgets about. Okay, so this is the dominant molecule in the ribosome. Okay, so if we remember ribosomal structure, we talked about this a few weeks ago. Remember, prokaryotes have this considered 70S structure, eukaryotes a little bit different, 80S. Remember, the S stands for Svedberg's, which is a measure of the sedimentation rate of the different pieces of ribosomal RNA, as well as the proteins that are associated with that. Okay, so for bacteria, the small subunit has this 16S ribosomal RNA piece along with a bunch of proteins. Okay, and then we have the large subunit that has a couple of other pieces of ribosomal RNA. Okay. Something else you should know about ribosomal RNA is that it can be catalytic. Okay, that means it can act like an enzyme. Okay, and when it acts like an enzyme, we have a special name for it. We call it ribozyme. Okay, so it's actually ribosomal RNA that can catalyze reactions. So when we get to translation, we'll talk about some of those reactions like peptide bond formation and cleaving of bonds, things like that. That's carried out by ribosomal RNA. Okay, and just a picture just so you appreciate the complexity. So this is, I'm not sure how they came up with this, but this is a 16S ribosomal RNA from a certain kind of bacteria called Dermis ponticus, I believe. So really complicated structure. Here, down here, the ribosomal RNA is the tan, and then the purple is the proteins that it's associated with. So that makes up the small subunit of ribosome in a species of bacteria. So again, just so you appreciate the complexity there. All right, so how does transcription take place? So this is the process of synthesizing RNA from DNA. And we break this down into three different events. So we talk about this in terms of initiation. And that's when our major enzyme involved here, Rna polymerase, will bind to a certain sequence of the DNA. And how well it binds depends on how likely that DNA is to get transcribed. Okay, and that happens at a place called the promoter region. Okay, so in bacteria, There's just one enzyme, RNA polymerase, that will make all the different kinds of RNA. In eukaryotes, there's a different polymerase for tRNA and a different polymerase for ribosomal RNA. So it gets a little bit more complicated, but just that one enzyme for bacteria. And then once it binds and starts transcribing the DNA, making RNA from the DNA, we call that elongation. Okay, we don't have to spend very much time there, but we're going to break this down further. And then events of termination, how that transcript gets stopped, how it stops making messenger or RNA from the DNA. Okay, so it's not just all of the DNA gets transcribed, it's only a piece of it. All right, so let's look a little bit more closely at that process. I think this is one area where your book really is not great. It just doesn't give enough detail, especially with initiation, to really make it very satisfying. So I've added in a couple other pictures that for me have helped me kind of make connections and understand what's really going on. So a little bit of outside of the book stuff here, especially for initiation. So remember, it's either one strand or the other of DNA that gets transcribed. Okay, and like I said, this RNA polymerase has to bind to this promoter region before it's going to start making that RNA transcript. So we'll talk about that in a minute. And it's adding these triphosphate nucleotides, ribonucleotides. Okay, so they carry the energy with them to polymerize, the polymerization reaction. So which strand gets transcribed? So in this case, it's drawn the lower strand of the DNA that gets transcribed. As to which one does, you can't really tell that by looking at it. The enzyme knows. It's whichever promoter that it sticks to. But there is some terminology that we use describing the strand that gets transcribed. Okay, so here, this antisense strand is the one that's being transcribed. Okay, we could also sometimes call that a negative sense. The other name we have for that is the template strand. Which should make sense. If you use a template for something, like if you've ever had to make a resume, you might use a template just so you kind of understand what you should be doing, like all the parts of it that you should make. So, you know, it's a guide that we use. So this DNA is the guide that's used to make that new messenger RNA. Okay, so if we drew in, I think this will kind of help understand a few base pairs there. Here, then this messenger RNA is going to be CGAU. Okay. The other strand, this one that didn't get transcribed, that's going to be called the sense strand, also known as the plus strand. And the other name we have for that is the coding strand. And that name maybe doesn't make quite as much sense. But if you write in maybe, you know, some, hey, what base pairs this opposite strand would be, it's going to be complementary to this DNA down here. So this would be CGAT. Okay, so what do you notice about CGAT and CGAU? Okay, they're the same, except RNA has uracil. Okay, so this coding strand is going to look like the new RNA. Okay, it's going to be the same basis. So it looks the same as the new code, I guess. You can think about it that way. Okay, so again, whichever strand is used, the cell knows which one it uses. And this messenger RNA, say this is messenger RNA, it's going to always make plus sense RNA. Okay, so use the negative strand of DNA when you're copying. That just means pluses and negatives are complementary. Okay, it's the plus sense of RNA that always gets read at the ribosomes. If the enzyme transcribed up here instead, it would be making the negative sense, and that's going to be encoding for a totally different protein. So there may be cases where both of those would make a protein that would be correct, but they would be different proteins. Maybe they're both useful for the cell, is what I mean. So I know that terminology is a little bit cumbersome, but when we get to talking about viruses, we have plus sense RNA viruses and negative sense RNA viruses. Okay, the plus sense ones, basically their RNA that they carry with them can just get read right away at our ribosomes. If it's a negative sense virus, then it has to make a plus sense. It has to synthesize that, have enzymes to do that, so that it can take over our ribosomes. So just kind of setting you up for that. But I know that's a little bit confusing. All right, so if we look at the major enzymes involved here, before we talk about what RNA polymerase is actually doing, I want to compare it to DNA polymerase 3, the enzyme in replication. A lot of questions that I can come up with for exams are trying to get you confused between the events of replication, transcription, and translation. So just want to try to keep those all kind of squared away in your mind. So RNA polymerase, this is the enzyme that's necessary to make new RNA. One interesting thing about it is that it can unzip the DNA itself. Where remember that DNA polymerase 3 in replication needed helicase to do that for it. Okay, so RNA polymerase does that itself. It doesn't need a primer. Okay, so kind of a self-sufficient enzyme, you could say. It can start de novo, we say that. So it can start transcribing messenger RNA kind of from nothing to start with, other than the DNA template. Of course, it has uracil instead of thymine over here. Proofreading is not as good. We'll say not as efficient. Okay, how often does that make an error? About 1 every 10,000 base pairs. Whoops. Will it make an error? All right, so not quite as good a fidelity, more mistakes made for making RNA, but again, that's okay. Probably next time the DNA is transcribed, it will be made correctly. So if it makes an error every once in a while, you could argue, well, maybe it's not that big a deal. Okay, and as we'll see, even more errors are made in translation. Okay, all right, so I think this figure is helpful to kind of see what's going on in those events of initiation. All right, so orient ourselves. Here's the DNA strand. And what we're zooming in on here is this region that we call the promoter. Okay, so that's a region of DNA. We call it upstream of the start site. So here's the gene up here that's going to get transcribed into messenger RNA. So this direction is upstream. So it's a series of maybe 35 or 40 base pair region upstream there. Here's the RNA polymerase. So this is a big enzyme that kind of encompasses, a whole bunch of base pairs. And the first thing to understand is that the more tightly that the RNA polymerase binds, the more likely the gene is to get transcribed. So there's some possibility of a little bit of regulation in there. So if this is a gene segment that's really important, this cell needs a whole lot of, the cell is going to want that RNA polymerase to bind really tightly there most of the time. So it will get transcribed most of the time. Maybe it's going to encode for a protein that isn't as needed as often. So maybe that RNA polymerase maybe will bind a little bit more loosely and only get transcribed, you know, a less percentage off or less often. Okay, so there's several things that will influence how tightly that RNA polymerase binds. Okay, one thing is this sigma factor. That's a lowercase sigma. I didn't realize till that long ago that Greek alphabet had upper and lowercase, but so that's a lowercase sigma. What is sigma factor? This is a protein that helps RNA polymerase. Bind to the promoter. Okay, so sigma factors come in and bind to that RNA polymerase right when it's trying to bind to that promoter region, and will make it more successful. There's different sigma factors that the cell uses. So again, it can kind of regulate how often that it's going to bind there really tightly. The other thing that's going to impact, especially how tightly it binds, is two regions in particular. And this is a -10 base pair region. We think that is so important that we've given another name for that in bacteria that's also known as the Pribnow box. I believe in eukaryotes, it's known as the Tata box, which kind of makes sense because it usually has a lot of thymines and adenines in it. But just in bacteria, we call that's kind of 5 or 6 base pairs, the Pribnow box. Okay, we think that sequence and the -35 base pair region are especially important for binding. Okay, so -10, -35, especially important for binding. Or RNA polymerase binding. Okay. Let's see if I said anything here. All right. So this next picture, this next graphic, again, helps. It looks confusing, but don't panic. It kind of helps for me pull some things together. Okay. So again, this is not from your book. What this is a sequence of the promoter region of a handful of different operons in E. coli. So sometimes, you know, we're drawing DNA, we draw a line, but we forget that's actually a sequence of base pairs. All right, so this is what we think the promoter sequence is for some different operons in E. coli. And if you remember, an operon is just, that just means it's a group of genes that are controlled by the same promoter. Okay, so remember we said in bacteria, messenger RNA is polycistronic, or it can be. All right, so in operons, that promoter will encode for several different proteins and not just one protein oftentimes. So there's actually, you know how many operons there are in E. coli? Like you talked about lac operon. We're going to mention one other one. there's like 800 different operons that have been discovered in E. coli. So here's just a few of them, what we think the sequence of that promoter is. So here's the -10 region in orange and the -35 region in blue. And they've gone through and they've been able to kind of understand, remember, those are the regions that are especially essential for RNA polymerase binding. They've come up with some consensus sequence. So they go through and, 69% of the time, this first base here is a T. And 79% of the time, the second base is a T. And so on and so forth. Like, don't memorize that sequence or those numbers. But they've come up with some consensus sequence, and they think that the more closer that -35 region is to that consensus sequence, then the more likely RNA polymerase will bind. Okay, so to some extent, the likelihood that the gene gets transcribed, that the RNA polymerase will bind, is written into the sequence itself. Okay, so let me sum that up. Genes with promoters that match consensus sequences are more likely to be transcribed. All right, so those are the ideal sequences for binding. All right, so enough about initiation. Once that RNA polymerase does bind tightly, then it can move down the DNA and start actually polymerizing the new RNA transcript. So here's your picture for elongation from your textbook. This is the sigma factor that you can see dropping off. Its job is done. Its job was just in getting initiation started. And if you look at the close-up here, you can see these triphosphate ribonucleotides adding on to the 3' end of the new strand. Okay, so the old strand, that's going to correspond to 5' and 3' of the old strand. Okay, and that's elongation. In eukaryotic elongation, there's a whole lot of other elongation factors that are involved there. It's much more simple for bacteria. Keep in mind that you can actually have lots of RNA polymerase coming in one after the other. So you don't have to wait for the gene to get transcribed once and then for that RNA polymerase to start over again. You can have many RNA polymerases and making lots of that same transcript. Where is this taking place? It's going to be taking place where the DNA is. So for bacteria, that's going to take place in the cytoplasm. Maybe the nucleoid region of the cytoplasm, but right there in cytoplasm. And then for eukaryotes, of course, the DNA is in the nucleus. Okay, so transcription would take place in the nucleus of eukaryotes. Okay, the prokaryotes right there in the cytoplasm. All right, how does transcription stop? There's two different ways that we think that happens, and we think that probably one happens about as often as the other. So, you know, half the time one way, half the time the other way. The one method is dependent on a protein called Rho. All right, so we call that Rho dependent. And Rho is a helicase enzyme. Okay, so helicase, remember, unzips 2 strands. So when the RNA gets transcribed, it's kind of weakly associated with that template just for a short period of time. That Rho protein, what it will do, will move along that newly transcribed messenger RNA and kind of break off these new associations all the way till the end of the transcript, and then the RNA polymerase dissociates from the DNA, and the helicase comes off, and now you have your new strand of RNA. To me, this is not very satisfying. Like, okay, how does the Rho know when to attach on? How does it do that? How does it know when the time is right to stop transcription? I've looked into this, and two things. Number one, it gets really complicated really quick. So we're not going to worry about it. And number two, there's a lot that we really don't quite understand how this happens. So for us in here, one way that transcription stops is with the Rho protein. And that's kind of as deep as we're going to go. The other method does not involve Rho protein. So sometimes we refer to it as Rho independent. Sometimes you hear this referred to as a sequence dependent termination, meaning it's just depends on the bases of DNA, that sequence of DNA that's being transcribed, or self-termination, meaning it terminates itself. Again, it doesn't need that Rho protein. So with this method, here we've got written in to that DNA sequence some palindromic sequences. Okay, so we've got if a word is a palindrome, it's the same forwards as back. So like the name Hannah is a palindrome, H-A-N-N-A-H. Same idea. It's a little bit different than that when we talk about palindromes in DNA, because these two sequences here, this is a little bit of a simplification because they are the same forwards as back. What they're referring to is kind of one stretch that then of DNA that's going to be complementary to another stretch when it's folded back on each other. Okay, so let me see. So for instance, if we had CCGGAA, and then a palindrome of that would be the complementary bases. T. So that's again the DNA sequence. Did I do that right? No, that's not what I meant to do. Sorry. So if it folded back on each other, it would be TTCCGG. Okay, because again, it's going to flop over this way. So those are considered palindromes. Neither here nor there, but the point is that the new transcript, what those palindromic sequences can kind of fold back on each other and make this hairpin loop. And the idea is that forces some mechanical stress that causes the transcript to dissociate from the DNA. Okay, so again, here they describe it as tension. Mechanical stress, that hairpin loop, which makes the RNA polymerase kind of release its grip from the DNA. In addition, you usually have a sequence here of A's and U's. They only have 2 hydrogen bonds, so they are going to dissociate a little bit easier than if you had C's and G's, which would have 3 hydrogen bonds. Okay, so the combination of that AU rich sequence as well as the hairpin loops, which can come about when there's palindromes, then that can just form some stress and make the transcript dissociate. Okay, so I want to compare for a second. Like I said, always assume that we're talking about processes in bacteria. But I want to compare for a second some differences with eukaryotes, because part of this I'm going to bring up later on when I talk about one of the drugs that's used to treat influenza, actually. Okay, so remember, I mentioned this before. Remember where transcription is taking place. For prokaryotes, it's in the cytoplasm. For eukaryotes, it's going to be in the nucleus. Again, where the DNA is. I mentioned also that eukaryotes, it's just a lot more complicated, this process. In eukaryotes, they've got different factors that have proteins that have to come in and regulate everything. And also in eukaryotes, there's some extra processing that happens to the messenger RNA after it's made. We have on the 5' end of the transcript, there's a guanine cap. And actually, this might actually get transcribed. It might not be added afterwards. So I might be presenting this a little bit different than what is actually occurred to me as I was preparing today. But there's a 5' guanine cap. So basically they are guanine bases that are just altered just a little bit. They're methylated and have a little different linkages. So that's put on one end of the transcript. And then on the 3' end, there's a polyadenylation. So again, on the 5' end, there's this kind of, sometimes we draw it like this guanine cap. And on the 3' end, there's a long series of adenines. Okay, so what this does, remember, because this is taking place in the nucleus, where is that transcript going after it leaves the nucleus? Okay, it's going to the ribosome, right? So it has to travel. So when you have this kind of buffer on the ends of the actual, what's going to get translated into protein, that's going to protect it. Okay, so these two things protect the messenger RNA as it travels to the ribosome. Okay, so we think that's really vital to just stabilize that messenger RNA transcript. And the third thing that happens, it's actually considered a pre-messenger RNA because it contains both introns and exons. Okay, the introns are the intervening sequences. I might have spelled that wrong. I don't think there's an I in there. Sorry. Intervening sequences. And then the exons are the expressed genes. Okay, so the introns get cut out, and the express genes, they are going to remain. Sorry, I didn't give a lot of room to write there at the bottom. Okay, so in bacteria, there's no introns and exons. But for eukaryotes, there's some kind of extra stuff that has to be cut out. So this is a nice picture from your book. Again, processing of eukaryotic messenger RNA. During transcription there, you can see there's both exons and introns. This kind of complex of enzymes called a spliceosome will work its way down the messenger RNA transcript and get rid of those introns. So you're just left with the expressed genes. Okay, and here we've got our 5' cap, our 3' polyadenylation. Okay, now it's ready to leave the nucleus. Got a date at the ribosome, okay, to get made into some protein. Okay, and just here's another picture that I found that from a different source. I kind of like the way that this is represented. So in the gene here, the little boxes, these are the exons, the thing that the sections that are really important. And then when you've got a pre-messenger RNA, the introns need to be taken out, but that can actually be done in slightly different ways. If it's only the introns that are taken out, you would probably get this messenger RNA. If it's cut like this on the bottom, you're going to get rid of that yellow box there and you would end up with this one. Or if it was done a little more creatively and part of this last exon was cut out, you would have a different sequence there. All three of these are going to give you a different sequence of amino acids and maybe different proteins. So sometimes we know that our cells do this on purpose. There's alternate ways to splice to give you different protein products, but sometimes our cells make mistakes and make errors in splicing. And we know that there are some diseases that are caused by errors in splicing. We think some neurodegenerative diseases like ALS, possibly Parkinson's, Duchenne's muscular dystrophy are all caused when splicing doesn't happen correctly. All right. All right. So let's have a practice question. I took this from an old exam, and it's probably one of those kind of wordier questions that just makes it a little bit more difficult because it's so wordy. So let me give you a second and see what you think, which one of those you would choose. So which one is true? Okay, any thoughts? What do you think? B. Hey, that's one. Okay. So you got to read multiple choice. You got to read it carefully. So first of all, which one is true, not which one is false, and make sure you're describing prokaryotes and also transcription, not translation. Right? That would be totally different. The -10 region is it's actually located upstream, not downstream. This is the opposite, a lot more errors in transcription than replication. This is an enzyme of replication, not transcription, so it's actually RNA polymerase. And we talked about in prokaryotes, there's not a nucleus in prokaryotes, so hopefully you didn't think it was that one. So good. I think it's good to pause and have a little practice. I think this is we're a couple minutes early, but I think this is a good time to stop. Then we won't, hopefully we'll keep transcription kind of separate in our minds from translation. We'll pick up there on Monday. Have a good weekend.